Today we will learn the chapter 9, Bacterial and Viral Genetic Systems. First we will learn the bacteria and the virus because they have a very important role in human society and the world ecosystem. After that we will learn about the bacteria genetic analysis and also gene exchanges through conjugation, transformation, and transduction. And also we will learn about the virus. So this slide will show the one example of the bacterium. This picture shows that women with leprosy we call it as nabyong. So nabyong is a disease caused by the bacterium which called the Mycobacterium leprae. So this is the nabyong. So 2013, a research group did a research. They found they isolated the leprae, the DNA from the ancient skeleton of medieval European. Comes from the UK, and there's also this skeleton comes from the UK, and this bone comes from the Sweden. Comes from Denmark. So they isolated M. leprae DNA from the ancient skeleton of medieval European with leprosy. After that, they did genome-wide comparison of the medieval and modern Mycobacterium leprae and found that there was some evolution of this bacterium. But today I will not cover in detail in this slide. Okay, so this is the one example of the bacterium evolution. So in this slide we will learn the first subject. Bacteria and viruses have an important role in human society and the world ecosystem. In chapter 2, we learned that the characteristics of the prokaryote. Prokaryote means that we summarize it again. Prokaryote is unicellular and no membrane-bound organelles, and the prokaryote DNA does not exist in highly ordered and packed arrangement. And usually prokaryote is made up of eubacteria and archaea. So again, we'll summarize the characteristics of the prokaryote again. All prokaryotes are unicellular and lack of membrane-bound nucleus and membrane-bound organelles. So all the prokaryote is composed of the eubacteria and archaea bacteria. Okay, eukaryote is composed of the animal, fungi, plant, and another species. Okay. In this slide, I will show you the characteristics of the bacteria. So bacteria is prokaryote. So usually the bacteria shows that exhibit diverse shape and size. This is the bacteria shape and the size. And also some of them is photosynthetic, especially the cyanobacteria is photosynthetic. And also usually the DNA replication, so this is bacteria chromosome. So usually the DNA replications occurs prior to the binary fission. So usually the DNA replication occurs prior to binary fission, and also because they only reproduced by binary fission, so it means that they can reproduce asexually. Because they can reproduce asexually means that maintain their characteristics from generation to generation. So they are the genetically pure strain. For example, the F1 generation is exactly the same as parent generation, and parent generation is exactly the same as grandparents, like this. And also, they are the haploid. For example, our human is diploid, but our sperm and egg is the haploid. But in the case of the bacteria, they have only one set of chromosomes. So they are haploid. This is very important. So in the bacteria, the recessive mutation are not masked. For example, in the diploid, if they have large R, small r, even though they have the small r, their phenotype will be the round. But in the case of the haploid, they will have only one chromosome set. So if they have the recessive mutation, small r, so their phenotype will be the ringed like this, not round. So because they are haploid, their recessive mutation are not masked. In this slide, I will summarize the advantage of using bacterial progenetic studies. Nowadays, many people use the bacterial progenetic studies. First of all, their reproduction is very rapid because they can divide it within 20 minutes. And also, many progenies are produced. And also, the important thing is that this one and this one, they have the haploid genome because their haploid genome allows all mutation to be expressed directly. So we already showed that because if they have the recessive mutation and the phenotype of the recessive mutation will be expressed directly. And also asexual reproduction simplifies the isolation of genetically pure strain, and also their growth is very easy and requires little spaces. And also nowadays the bacteria can be genetically engineered to produce substance of commercial value. For example, nowadays the insulin is produced in the bacteria. Okay, so in this slide, I will explain where bacteria are grown. So usually the bacteria are grown in the liquid medium, or they can be grown on the solid medium. So this is the agar plate. The plate is the space is very solid, so they can grow in the liquid medium and on the solid medium. And also we can divide the bacteria into the prototrophs and the auxotrophs. In prototroph, in Korean we call it as the 자가 영양세균. 자가 영양세균 means that on microorganism capable of synthesizing all its metabolites from inorganic material. How about auxotrophs? In Korean, we call it as 영양요구성 세균. 영양요구성 세균 means that a mutant microorganism that has lost their ability to synthesize an organic compound required for its growth. This is prototrophs. For example, they have the essential gene, they have all of the essential gene for growth, but the auxotrophs, due to some gene deletion or there is some point mutation, so this microorganism that has lost their ability to synthesize an organic compound which is required for its growth, so it means that they need additional nutrient. It means that we call it as 영양 요구성 세균 or the auxotrophs. So prototrophs, they don't need they don't need additional nutrient for their growth because they can capable of synthesize all its metabolites from inorganic material. Now I will explain the techniques for the study of bacteria. First of all, we have to know some terminology. First one is minimum medium. In Korean, we call it as 채소배지. 채소배지 means that a cultural medium that contains the minimal necessities for the growth of the prototrophic bacteria. So it means that once again, they contain minimal necessities for the growth of the prototrophic bacteria. How about complete medium? In Korean, we call it as a 완전배지. So a culture medium that contains all of the growth requirement for the growth of all bacteria, including the auxotrophic bacteria. So this is the complete medium. So this is the technique, one technique for the study of the bacteria. For example, this bacteria culture contains the prototrope and the auxotroph. The prototrope is the one is genotype is leucine plus, means that this is prototrope, and leucine minus means that auxotroph. So they need leucine for survival. So this is a bacteria culture which contains the prototrope and auxotroph. So now we plate the bacteria on media containing the leucine. So it means that both leucine plus and leucine minus colonies can grow. So this is like this. And after that, so now we have to replicate the plate. So now we use the velvet space. So this is a replica. We call it as a replica. They is composed of the velvet space. So by pressing a velvet space onto plate, so after that their colony can be go to in the velvet space like this. So cells adhere to the velvet. So in one cases, this is medium with lacking the leucine. So this is the and also this is the medium with the leucine. So this is the minimal medium, and this medium will be the complete medium with the leucine. So after that, we can press onto new petri plate. So cells from each colonies are transferred to the new plate, and the new plate is like this. So in these cases, in complete medium, okay, all of the both leucine plus and leucine minus colonies can grow here and there, but in the minimum medium without the leucine, you can see that some of the colonies are missing. So here, all of colonies existed exactly the same as this one, but only this colony is missing. So it means that leucine auxotrophs are recovered from the colonies growing on the supplemented medium, means growing on the complete medium. So this colony is leucine auxotrophs. The genotype will be the leucine minus. So after that, we just culture this one for the further studies. So conclusion was that colonies that grows only on the complete medium, for example, this colony only grows in the complete medium, has a mutation in a gene that encodes the synthesis of the essential nutrient. In this case is leucine. Okay. This slide I will explain the bacterial genome. So bacterial genome is composed of the chromosome and plasmid. So this is bacterial chromosome and this is bacterial plasmid. So bacterial chromosome is mostly single. It means that they are haploid, and also they are circular DNA molecules. And how about plasmid? plasmid is also composed of two types. One is the small circular DNA, and another is the episome. Let's start from the small circular DNA. So this is the small circular DNAs. They have the origin of replication. So replication in a plasmid begins at the origin of replication, and after that the strand is separate and replication takes place in both directions. Eventually, they will produce two circular molecules like this. How about the episome? So episome is this is episome. So episome we call it as the F plasmid. For example, the episome contains the gene that regulate plasmid transfer into a bacterial cell. So this is the genes that regulate plasmid transfer into other cell. For example, this is the F plasmid episome, and usually they conjugate to each other. At that, the plasmid DNA can be transferred into the other bacterial cells. So actually, this gene can control all of the process. How about, and also the episome also has the origin of replication, so it can regulate DNA plasmid replication, and also they have the IS2 or IS3, and this is the sequence is regulate insertion into the bacterial chromosome. So the F plasmid can be integrated into the bacterial genome due to they have the IS1 and IS2 and IS3. But the small circular DNA cannot be integrated to the bacterial genome or cannot be transferred into the other bacterial cell. But episome contains the plasmid contain the gene that regulate plasmid transfer, and they have gene which can regulate insertion into a bacterial chromosome. But both plasmid has the gene which control the plasmid replication. So this small circular DNA has the origin of replication, and the episome also has the origin of replication. Okay, this is concept check 1. Which statement is true or plasmid? So A is they are composed of the RNA. B is they normally exist outside of a bacterial cell, and C is they possesses only a single strand of DNA, or D is they replicate independently of a bacterial chromosome. The answer is D, because the small circular DNA, they have the origin of replication, and the episome also they have the origin of replication. So the answer is D. In this slide, I will explain the third subject, bacteria exchange genes through conjugation and transformation and transduction. In this slide, we will learn the gene transfer in bacteria. There are three types of the gene transfer. First one is conjugation, the other one is transformation, final one is transduction. Conjugation means that direct transfer of DNA from one bacterium to another. For example, this is the conjugation. For example, this is donor cell and recipient cell. Usually, the cytoplasmic bridge forms between donor and recipient cell. After that, the DNA is replicated and transferred from one cell to the other. So this is the transferred DNA replicates. After that, crossover is happen, and that it can create the recombinant chromosomes like this. How about transformation? Transformation is usually the bacteria is taken off free DNA from outside. So this is the DNA fragment. So for example, naked DNA is taken up by the recipient cell. After that, the crossover is happened. After that, it can create the recombinant chromosomes like this. How about the transduction? Transduction means that bacterial virus, we call it as the bacteriophagy. Bacteriophages takes DNA from one bacterium to another. So this is the transduction. Usually the bacterial virus, we call it as the bacterial virus. We call it as bacteriophagy attached to the bacterial cell. After that, it can inject its DNA, and after that, the bacterial DNA can be integrated into the bacterial genome. After that, they can replicate and take up of the bacterial DNA, and after that, finally, they will do bacterial cell lysis. So, and after that, this is the new virus, but this virus has the bacterial DNAs like this. They have the bacterial DNAs like this. Okay, and this virus is also impact another new bacterium. After that, because this virus carries bacterial DNA with it, after that, the crossing over happen. After that, this bacteria can create no recombinant chromosomes like this. So usually the transduction, the bacterial virus, we call it as bacteriophagy, takes the DNA from one bacterium to another. So this is, we call it as transduction. So this is a concept track 2. Which process of DNA transfer in bacteria require a virus? Conjugation, transformation, transduction. Which one? The answer is C, transduction. In this slide, I will explain the genetic exchange or gene exchange in bacterial cell. The question was that, do bacteria exchange genetic information? To answer the question, Lederberg and Tatum did experiment. Before I explain the details in the experiment, I will explain some terminology. F plus cell and F minus cell. F plus cell means that donor cell containing F factor. F factor means that F factor is located in the episome. So episome, this is episome, and this location is the F factor. This F factor is regulate plasmid transfer into another bacterial cells like this. So F plus cell means that donor cells containing F factor, and the F minus cell means that recipient cell lacking the F factor. And how about the sex pillars? Between F plus and F minus cell, usually there is a connection tube is made, so we call it as sex pillars, and this is connection tube which connect between the F plus and F minus cell. So okay, this is the experiment. Okay, Y10 strain means that this bacterial strain is the auxotrophic. So for example, this is a threonine minus and leucine minus and Ti minus, means that they cannot synthesize threonine and leucine and Ti. But this strain can synthesize biotin, naphtophenylalanine, and cysteine. How about the Y24? Y24 can synthesize the threonine and leucine and dye, but they cannot synthesize the biotin and phenylalanine and cysteine. So, okay, so so Y10 is auxotropes and Y24 is auxotropes. So neither auxotrope strain can grow on the minimal mediums like this, because they are not prototropes, they are auxotropes. So neither the auxotropes, the Y10 or auxotropes Y24 can grow on minimal mediums like this. So now is people mixed Y10 and Y24 together, and or at that we can grow in the minimal grow on the minimal medium, and found that there are several colonies show up, and people found that this colony show that they have the threonine positive and also leucine positive and cysteine positive, biotin positive, phenylalanine positive, cysteine positive. So it means that because genetic recombination has taken place and the bacterial cell synthesized all necessary nutrients. So yes, the conclusion was yes, genetic exchange and recombination took place between two mutant strains. For example, maybe if this is the F plus cell, or if this is the F minus cell, so maybe the gene can be transferred to this F minus cell. At that, some recombination happened. At that, they will make the recombinant chromosome. At that, so this colony which has the recombinant chromosome can survive can grow in the minimal mediums like this. So the conclusion was a genetic exchange and recombination takes place between two mutant cells. So it means that bacteria can exchange genetic information. People see the experiment which was conducted by Lederberg and Tatum. After people has another question. The question is, how did the genetic exchange seen in the Lederberg and Tatum experiment take place? To answer this question, they did the YouTube experiment. So one is that this is the one is auxotropic strain A, and the other strain is also auxotropic strain B, is like this. Okay, so at that Davis did the YouTube experiment, and two auxotropic strains were separated by a filter that allows the mixing of the medium, only mixing of the medium, but not bacteria. At that, people did the experiment and found that no prototropic bacteria were produced from this experiment. So this auxotropic strain A is they cannot grow in the minimal medium, and also this auxotropic strain B also cannot grow in the minimal medium. And also this strain A and strain B are mixed each other, but they are separated by the filter. In these cases, there were no prototropic bacteria. It means that genetic exchanges require the direct contact between the bacterial cells. It's like this. So this is usually the F plus and F minus cell. They need sex pillars which can connect F plus and F minus cells like this. So the conclusion was genetic exchanges requires direct contact between the bacterial cell, especially F plus and F minus cells. In this slide, I will explain some details in the conjugation. The detail is that direct transfer via connection tube is a one-way traffic from donor cell to recipient cells. So it is not reciprocal exchange of genetic information. Reciprocal means that 상호간, it is not reciprocal exchange of genetic information. For example, let's start from the F plus and F minus cell. So F plus cell has the F factor, has the episome. So this is the donor bacterium. The other don't have the F factor. So this is the recipient bacterium. So usually during conjugation, the cytoplasmic connection is a connection tube is formed. After that, one strand of the F factor is nicked and at the origin of transfer and can be separated. After that, the 5' end of the nicked DNA is passed to the recipient cell, after where the single strand is replicated, finally producing a double-stranded copy of the F plasmid. So this was F plus and F minus cell. After conjugation, still this is F plus and F minus, F plus, F minus, F plus, F minus. Now with the F minus cell becomes F plus cell because they have the F factor. So conjugation, the details in conjugation is direct transfer via connection tube is one-way traffic from donor to recipient cell. It is not reciprocal exchange of genetic information. Explained another details in conjugation. So we have to know some terminology. HFR cells. HFR cell means a high frequency strain, means that in Korean we call it as 고빈도 제조합. This means that this cell is donor cells with F factor is integrated into donor bacterial chromosome. So this is actually the F plus cell, and this F plus cell has bacterial chromosome. Also they have the F factor, F plasmid. So this F factor is integrated into the bacterial chromosome, and at that this become the HFR cells. So this cell can be function as donor cell. So here this is donor cell, and this can be the F minus cell because they don't have the F factor. After that, usually the bacterial gene can be transferred from HFR cell to F minus cell through conjugation. After that, HFR chromosome, this is HFR chromosome. This is F factor can go to the F factor plus bacterial genome can be transferred to the F minus cells. After that, usually in these cases, crossing over takes place between donated HFR chromosome and also the original chromosome of the F minus cell. So usually in here, the green chromosome segment comes from the donor's HFR cell, and this black segment is located in the F minus cell. So after recombination is happened, crossing over happens, lead to the recombination is over earlier. So this F minus cell have the green segment, the bright. So the crossing over may lead to the recombination of earlier, the bright green in the place of the black segments. So after that, this linear chromosome is degraded. After that, this cell still become the F minus cell, but crossing over happens. So this is new recombination of the alleles. Okay. While I explain another details in conjugation. First of all, we have to know F prime cell. F prime cell contains F plasmid carrying some bacterial genes. So this is the one example. So it initially start from the HFR gene. So in HFR cell is the F factor is integrated into the bacterial cells. Sometimes crossing overs happens within the HFR chromosome, and that sometimes the F factor is excised from the bacterial chromosome. But in these cases, it carries F factor carries some bacterial genes. In this case, is the lactose gene. So actually, the lactose gene is located in the bacterial chromosome, but the F prime cell has the F plasmid carrying some bacterial gene. So this is the F prime cell, especially in this case is the lactose gene. So this is the F prime cell function as a donor cell because they have the F factor, and the F minus cell function as a recipient cell. So during conjugation, the F factor with the RAP gene is transferred to the F minus cells, and after that it can still produce the F prime cell, which has the F plasmid carrying some bacterial gene. So in this case is the producing partial diploid cell with two copies of RAC gene and the RAC gene. So in this case, we call it as a F prime cell, but we also call it as a merozygote. Merozygote means that partial diploid bacterial cell. Actually, the bacterial cell is haploid, but in this case is partial diploid bacterial cell carrying the F plasmid containing F plasmid carrying some bacterial gene. So this is, we call it as F prime cell, especially merozygote. So in this slide, the summarize the characteristics we correlate with different type of F factor. First, we already know the F prime cell, F plus cell, and the F minus cell. F plus cell, in F plus cell, the F factor is present as a separate circular plasmid, actually in the episome. So F factor is present as a separate circular plasmid, so the F plus cell function as a donor cell. How about F minus cell? So there is no F factor, so in this case is F minus cell function as a recipient cell. How about HFR cell? HFR cell, F factor is present, but F factor is integrated into bacterial chromosome. So usually the HFR cell function as a high frequency donor. So the donor, the role as donor is the higher than the F plus cell. So how about the F prime cell? F prime cell is the F factor is present as a separate circular plasmid, both carrying some bacterial gene. So also F prime cell function as the donor cell. Okay, so in this table 3.3 summarize the result of conjugation between cells with a different F factor. How about let's start from F plus cell and F minus cell. This is F plus cell and F minus cell. After conjugation, they will become the two F plus cell and F plus cell. So F minus cell becomes F plus cell. How about the HAPR with the F minus cell? So usually still after finish the conjugation, still one HAPR cell and one F minus cell. But in F minus cell, so the crossing over happened. There was some recombination in the F minus cell. But still this cell is the F minus cell. How about F prime with F minus cell? In this cases, this is F prime cell with F minus cell. In this case, the two cells become the F prime cell. So actually, the F minus cell becomes F prime cell. It's like this. So this is concept check 3. The conjugation between the F plus cell and F minus cell usually result in which of the following? The following is the 2F plus cell. The answer is A. In this slide, I will explain how to map the bacterial genes. So to map the bacterial gene, we usually use the interrupted conjugation. For the interrupted conjugation, we used HAPR cells and also F minus cell. So HAPR cell with the genotype streptomycin sensitive. So they don't have the streptomycin resistance, and also threonine positive and leucine positive. They also they have the Ag resistance, means that azithromycin resistance. And also they have a tone resistance. Tone resistance means that bacteriophage T1 resistance. It means that if they have a tone resistance, bacteriophage T1 cannot infect this bacteria. And also they have a lactose positive and they have the galactose positive. How about the F minus cell? The genotype of the F minus cell is the genotype is the streptomycin resistance. Instead of the streptomycin sensitive, they have a streptomycin resistance. And instead of a threonine positive, they have threonine negative. And also, instead of a leucine positive, they have a leucine negative. And also, instead of the azithromycin resistance, they have the azithromycin sensitive. And also instead of the tone resistance, they have a tone sensitive gene. And also lactose positive, they have a lactose negative. And galactose positive, they have a galactose negative. Okay, so after that, we used the time interval. For example, so after we put the HAPR cells and F minus cells together, after that 8 minutes later, or 10 minutes later, and 16 minutes later, and 25 minutes later, and conjugation was interrupted at regular interval. How to interrupt the conjugation? Usually, for example, we usually use the sonication. So after 8 minutes later, we can do the sonication. So, or after 10 minutes later, we can do sonication, or 16 minutes later, another group in 16 minutes later, we can do sonication. In this case, the connection tube is disconnected. Or 25 minutes later, we can do the sonication. So after that, this is the graph result. This is a percentage of cell displaying particular trait. How about the recipient cell actually was azithromycin sensitive, but around 8 minutes later, they have the azithromycin resistance gene. How about tone? Bacteriophage T1 resist. How about tone? Actually, the recipient cell they are tone sensitive, but around 10 minutes later, they should display the tone resistance phenotypes like this. How about the lactose positive? Actually, they were lactose negative, but about 15 or 16 minutes later, they show that lactose positive phenotype. How about the galactose? So initially the F minus cell, they are galactose negative, but around 25 minutes later, they have the galactose positive phenotype. So this x-axis means the time after start of the conjugation between HFR and F minus cell. So the conclusion means that transfer time indicate the order and the relative distance between gene and can be used to construct a genetic map. So for example, azithromycin resistance show up the first, so maybe azithromycin, after that there will be the tone, after that there will be the lactose, after that there will be the galactose genes. So this is the order. So based on this experiment, we can find the order of gene, the AZ, tone, log, galactose. And also we can determine the relative distance between gene. Maybe between the AZ and tone, maybe there will be the 1 minute distance. So maybe distance will be the 1 minute, or the relative value is 1. How about tone and log? Between tone and log, there was around 8 minutes difference. So maybe the distance between the tone and the log will be the 8. About the relative value will be the 8. How about between the log and gar? The time difference is about 8 minutes, means that the distance between log and the galactose will be around the 8, is like this. This slide, I will explain some details in the gene mapping of the bacterial genome. So we have to know some important characteristics of the F factor. So F factor has orientation. So this orientation enables directional transfer. Okay? For example, different FR strains, HFR1 and HFR5 have the F factor, they have F factor integrated into the bacterial genome chromosome at different sites and in different orientation. So F factor is integrated at different location. For example, in this case, F factor is integrated between the leucine and the RZ gene. How about in these cases? In this case, F factor is integrated between di and histidine gene. And also the orientation is, in this case, orientation is like this. How about in this case? The orientation is like this. So different HLPAR strain have the F factor integrated into the bacterial chromosome at different site and also in different orientation. But important thing is that gene transfer usually begins within F factor. So it means that in this case, in this HFR1 cases, usually the transfer always begins within F, and the orientation of the F determines the direction of transfer. So the orientation is like this. Okay, that in HFR1, F1 is integrated between and leucine and RZ gene. At that, SOR the genes are transferred beginning from the leucine, threonine, dihistidine, GAR, LAP, PRO, and RG is like this. How about in these cases? Okay, in this case, the F factor is integrated between the di and histidine, and the orientation is also like this. In this case, the gene is transferred beginning from di, threonine, leucine, RG, proline, LAC, GAR, histidine. So it means that the orientation and the position of F determine the direction and the starting point of the gene transfer. So this is important criteria we have to know when we do gene mapping in bacterial genome. Okay? In this slide, we will learn some detailed information of transformation. In previous slide, we already learned the transformation. The transformation is bacterium takes up free DNA from the medium. After that, the recombination happens. After that, the fragment is integrated into the bacterial genome. So this is the transformation. So now we will see some detailed information of transformation. So usually in the media, there is a double-stranded fragment of DNA is located here. After usually among the two-stranded DNA, only the one strand of the DNA is entered the cell and the other is hydrolyzed. After that, single-stranded fragments pairs with the bacterial chromosome and the recombination takes place. After that, this single-stranded fragment is integrated, recombined with the bacterial genome. So recombination takes place between the introduced gene and the bacterial chromosome. And also remained one, and this remainder DNA is degraded. After that, this recombination happens, and when the cell replicates and divides, one of the resulting cell is transformed, means that this cell has the bacterial genome which has a recombination. The other cell is a non-transformed, but the other cell don't have don't have the introduced the gene. So this is a non-transformed. So this is the detailed information of transformation. Slide, I will explain some important terminology in transformation. First of the recipient cell. Recipient cell means the cells that takes up the DNA. So this is recipient cell. For example, the genotype of recipient cell is A- and B- and C-. How about the donor cell? This is a donor cell. The donor cell means that the cells which provide the DNA. So usually the apt donor cell provide the DNA in the outside. So DNA is from donor cell is usually fragmented like this. And also the fragmented DNA are taken up by the recipient cell, like this. Okay. How about the transformant? Transformant means that cells that receive genetic material. So cells that receive genetic material. How about start from this one? In this case, this cell received A plus DNA fragment. After entering the cell, the DNA becomes incorporated into the bacterial chromosome through crossing over. So this transformant, the genotype becomes A plus, B minus, C minus. So transformant means that cells that receive genetic material. How about in these cases? In this case, is the this cell uptake the B plus DNA fragment. After that, the B plus DNA fragment is integrated into the bacterial chromosome by crossing over. How about in these cases? In these cases, they receive the C plus. After C plus is integrated into the bacterial chromosome by crossing over. How about the co-transformed? So co-transformed means that cells that are transformed by two or more genes. So cells transformed by two or more genes. In this case, is A plus B plus is transformed together. So this is co-transformed. So in the case of co-transformed, we can use transformation can be used to map the bacterial gene. Okay, in this case, we'll see that in donor cell, this is the gene A is located here, gene B plus is located here, and C plus is located here. So it means that the distance between A and B is very close to each other. How about the distance between A and C? A little bit far away from here, okay? How about B and C? B and C is the distance is far away. So, for example, the genes that are close to one another on the chromosome are more likely to be present on the same fragment. So, in this case, the genes A and B they are close to each other. So it means that the genes A and B are likely to be present on the same DNA fragment. And after that, it can be recombined together. So it means that the rate of co-transformation is inversely proportional to distance between genes. So if the rate of co-transformation between A and B is high, means that the distance between A and B is very close to each other. How about if the rate of co-transformation between the rate of co-transformation of A and B is very low, means that the distance between A and B is far away from here, far away. Distance is a lot. So it means that by using the rate of the co-transformation, so we can determine the map distance between A and B, because the rate of co-transformation is inversely proportional to the distance between gene A and B. Okay, genes. Okay. From this slide, I will explain the final subject. Viruses are simple replicating systems amenable to genetic analysis. Okay, let's start from the virus. The basic unit of the virus is composed of the replicating structure, DNA or RNA, and they also have protein coat, which is we call it as capsid protein. So this is a basic structure of the virus. So after that, DNA or RNA is encircled by the capsid protein, so they will make the nuclear capsid. So this is one example of nuclear capsid. So capsid protein is the encircle the DNA or RNA, so they will make this kind of structure. Or how about the tobacco mosaic virus? This nucleic acid is located inside and the capsid protein is outside like this. And also, there is the two types of virus. One is naked virus. Naked virus means that they only have the nuclear capsid. They don't have any envelope. How about the envelope virus? So in addition to nuclear capsid, they have the lipid membrane and glycoproteins, and this lipid membrane with glycoprotein is envelop the virus. So the nuclear capsid is inside the envelope. So this is the enveloped virus. This slide I will explain the bacterial phases. Bacterial phase is the species of virus, the virus that can infect the bacteria. We specially call it as a bacterial phase. So bacterial phase is composed of two types. One is the virulent phase and the other is temperate phase. Okay, let's start from the virulent phase. So, so bacterial phase usually bind to the bacterium. After that, the phage DNA is enter the host cells like this. After phage DNA is enter the host cell, usually the host DNA is digested. After that, phage DNA replicates by using host cell machinery. After that, the host cell transcribes and translates the phage DNA, and after that producing the phage proteins for their capsid like this. After that, assembly of new phage is complete, and after that the phage encoded enzyme causes the cell to lyse. After that, new phage are released and start to the cell cycles again. So this is the lytic cycle. So virulent phages reproduce through this lytic cycle. So they always kill the host cell. So this is plaque. So in the LB plate, we usually we add the bacteria. So they will make the lawn of the bacteria. After that, we add the virulent phages. So virulent phase is in this cases, in this location, there was the virulent phase. So it kills the host cells, so they will make the clear patches like this. So plaque, we call it this as the plaque. Plaque are clear patches of the lysed cell on the lawn of the bacteria. So this is the virulent phase. How about the temperate phase? So temperate phase means that phage DNA. So in this case, phage DNA. This phage DNA is integrated into the bacterial chromosome and becomes prophase. So phage DNA integrated into the bacterial chromosome where it remains as inactive prophase. So we call it as prophase. So this page is the inactive phase. We call it as temperate phase or the prophase. So usually the prophase is replicated as the part of the bacterial chromosome is replicated, and these replications can continue through many cell division. So after one division, they will make the two. After second division, they will two become the 4, and 4 becomes 8, something like that. But at some time point, this prophase is may separate from the chromosome, and the cell will enter the lytic cycles like this. Okay, so this is this cycle, we call it as a lysogenic cycle. In this case is temperate phase, or another name is prophase. Prophase is involved in the lysogenic cycle. In this slide, I will explain the Lederberg and the Ginder experiment. So previously we found that based on the Davis YouTube experiment, we found that genetic exchanges requires direct contact between the bacterial cell, between the F plus and F minus cell. That is, they need direct contact. But now is the question is does genetic exchanges between bacteria always require cell-to-cell contact? So this is experiment. This is question. To answer the question, the Lederberg and Ginder did experiment. So one group they used the oxotropic strain, which one is this strain is phenylalanine positive and tryptophan positive and tyrosine positive, but methionine negative and histidine negative. How about this strain? This strain is also oxotropic strain, which has phenylalanine negative and tryptophan negative, tyrosine negative, but methionine positive and histidine positive. After that two oxotropic strain is mixed together, after that plated on the minimal medium. So finally, we found that some prototropic colonies were obtained. So the genotype was the phenylalanine positive, tryptophan positive, tyrosine positive, and also methionine histidine positive. So in the other experiment, people did use the same oxotropic strain. After that, put the two strain in the Davis YouTube. So in Davis YouTube is in which the strains were separated by filter with the pore too small for the bacteria to pass through. Okay, after that, the colony from this tube they plated on the minimal medium, and also the colonies from this tube they plated on the minimal medium, and found that from this tube there was no colony, but from this tube people found that prototropic colonies were obtained only one side of the tube. And also genotype is also phenylalanine and tryptophan, tyrosine, methionine, histidine positive. How can this phenomenon happen? How? So because of there was a filter, so there was genetic exchanges did not take place through conjugation. But how? So maybe the Ph.D. is the agent of transfer. So Ph.D. did the Whitic cycle and the lysogenic cycle. So maybe Ph.D. is impact this bacteria after during their Whitic cycle and the lysogenic cycle. They can transfer the genetic material from here to here. After that, in these colonies, they receive the bacterial genetic information from Ph.D. So finally, there was the recombination. So finally, they will make the prototropic colonies. So in this case, is Ph.D. Ph.D. is too small, so they can pass through this filter. So the conclusion was genetic exchange did not take place through conjugation. Maybe Ph.D. was later shown to be the agent of transfer. Okay, I will explain some detailed information of transduction. So previously, our conclusion was the Ph.D. is the agent of transfer, but I will not explain the detailed information of gene transfer by the Ph.D. Now I will explain some details information how bacteriophage transfers bacterial gene to another bacterium. So this is the transduction. Transduction means that bacteriophages transfers bacterial gene to another bacterium. So usually the transductions occurs between the bacteria of same or closely related species. Okay, now let's see the detailed information. First are the bacteria infected with phage. So this is phage DNA. This is purple color, and the donor bacterium, the color, the chromosome, the color is the red, is like this. After phage DNA is inserted into the donor bacterium, so usually the bacterial chromosome is fragmented. So the chromosome is fragmented. At that, some of the bacterial gene become incorporated into the phage DNA. So at that, they become incorporated into the new phage. So this phage, we call it as the transducing phage. So they can contain the bacterial genes. And this is the normal phage. They only contain the phage DNA. So after cell lysis, so it can release the transducing phages. So in some cases, this transducing phage impacted the recipient cell. So if the phage transfer bacterial gene to another bacterium, usually the recombination take place, and at that they finally produced transduced bacterial cell. So we call it as a transductant. So usually the recombination take place and produces or transduced bacterial cells. The general name is transductant. Okay, so this is the generalized transduction, the mechanism. So in this slide, I will explain how to use the transduction when we do the gene mapping. So, okay, I will explain some of the principle of the transduction. So, okay, so this is the donor strain of the bacteria. This is a donor strain of bacteria. Their genotype is A plus and B plus and C plus. This donor bacteria is infected with bacteriophage. After that, the bacterial chromosome can be the breakdown, and also some of the bacterial gene can be integrated into the some of the progeniphages. In these cases, the A plus, the gene is integrated into the progeniphage, the genome of the progeniphage. So in this case, the B plus, the gene B plus is integrated into the genome of the progeniphage. How about in this case? In this case, the C plus gene is integrated into the genome of the progeniphage. Okay. So in this case is so A plus, and also the bacterial gene can be inserted into this recipient bacteria. This recipient bacteria is genotype is A minus and B minus C minus. So in these cases, A plus can be recombined to the bacterial genome. After that, they will have some recombinant. The genotype is A plus and B minus and C minus. How about in these cases? And B plus can be after this phage is impacted the recipient bacteria, and B plus gene can be transferred to the inside of the recipient bacteria. So in this case they have the B plus. So in this case the recombination is take over between here and here. So the B plus can be integrated into the recipient genome. So it will make the A minus, B plus, and C minus. How about in these cases? The C plus gene is integrated into the genome of the progeny's progeny, except that the DNA of the progeny can be inserted into the recipient bacteria. So in this case, they have a C plus gene. So the combination takes place between this one and this one. So some recombinant will be made. The genotype will be the A minus, B minus, and C plus. So in this case, we call it as a single transductant. How about in these cases? So in these cases, A plus and B plus gene can be integrated into the genome of this progeny. After that, the progeny is impact the recipient strain of bacteria, which has the genotypes A minus, B minus, C minus. After this, the A plus, B plus can be go inside of the recipient strain of bacteria. After that, the recombination can be happened. After that, this A plus, B plus can be integrated into the recipient the recipient strain of bacteria. So final genotype will be the A plus, B plus, and C minus. So it means that genes located close to one another are more likely to be co-transduced. In this case, the gene A and B they are close to each other, but how about the A and C? They are far away from each other. How about B and C? The B and C is also far away from each other. So the conclusion is the genes located close to one another are more likely to be co-transduced. So it means that the rate of co-transduction is inversely proportional to distance between genes. So if the rate of co-transduction is high, means that the gene location between A and B is very close to each other. If the rate of co-transduction is very low, it means that the distance between the A and B is very far away from each other. So this is we use the general transduction when we do the gene mapping. So you use this principle. The rate of the co-transduction is inversely proportional to the distance between genes. So, okay, this is concept chapter. So in gene mapping experiment using generalized transduction, bacterial genes that are co-transduced are far apart on the bacterial chromosome, or on different bacterial chromosome, or close together on the bacterial chromosome, or are on a plasmid. What's the answer? The answer will be the C. So usually the bacterial gene that are co-transduced are close together on the bacterial chromosome. So this one is the answer is C. In this slide, I will show you how to do gene mapping in the fuzzy. So now is the question was how can we distinguish how can we determine the position of gene on a fuzzy chromosome? How about in this experiment, the phagy has one phagy has they have their genome as H plus and R minus, and the other phages they have H minus and instead they have R plus. So E. coli cell was infected with two different strains of T2 phages. One is so these phages impact the bacterium after that they released their DNA. So their DNA is this is their genotype H plus and R minus. How about this bacteriophagy? This bacteriophagy also impact this bacterium, so they released their chromosome or their DNA. Their DNA is genotype is H minus R plus. So usually sometimes the crossing over between two viral chromosomes happens. So if there is a crossing over happens, so they will make the H plus and R plus. The other one they will make H minus and R minus. And also some viral chromosome did not cross over, resulting in the non-recombinant progeny. This is also non-recombinant progeny, and this is also non-recombinant progeny. So in these cases, this non-recombinant progeny produces in this case is very large and cloudy plaque. But how about in this case, this non-recombinant progeny produces the very clear. This one is very clear, but very small plaques like this. Okay, but how about after this recombination? How about this progeny, recombinant progeny, produces cloudy but small plaque. How about this one? This recombinant progeny produced clear but large plaques like this. So this is a table 9.4 which summarizes the progeny phases produced from this crosses, produced from this bacteriophagy and this bacteriophagy. Okay, so in this case is clear and small, and cloudy and large. The genotype is like this. They are non-recombinant, and cloudy and small, and clear and large. This is the recombinant. So after that, progeny phagy were then plated on a mixture of E. coli, and that we can count it. After that, we can count it the which one is the clear and small, and which one is a cloudy and large, and which one is a cloudy and small, and which one is a clear and large. So okay, in this case is so H minus R plus. So in this case it means that clear and small. So when they counted, so clear and small, they found 42 plaque. How about this one H plus R minus? H plus R minus means that cloudy and large. Cloudy and large, we can count it and we found that 34 plaque. How about H plus R plus means that cloudy and small. So cloudy and small. So we can calculate. We can count how many cloudy and small plaque is there. So after we found the 12. How about H minus R minus means that clear and large. So in clear and large, we can count it and find that there was 12 plaques. So it means that this is the non-recombinant, and this is the recombinant. So the RF recombination frequency is total plug. Total plug is that 42 plus 37, 34 plus 12 plus 12, and after that the recombination blocker means that 12 plus 12. So it means that total will be the 24%. After that, just multiply 100, and the recombination frequency will be the 24%. So conclusion was the recombination frequency indicate that the distance between the H and R gene is 24%. Okay, so the recombinant frequency means that this is the 0.24, and the recombination frequency 0.24 indicates that the distance between H and R will be around the relative 24%, something like that. So this is the how we do gene mapping in the pharmacy. So just use the recombination frequency. The how to calculate recombination frequency is based on this calculation. So total plugger or per recombinant pluggers. So now we know the phenotype of recombinant pluggers. So one is the cloudy and small, the other is the clear and large. So the total number is like this. So the recombinance frequency will be the 0.24, and the recombinance frequency indicates that the distance between H and R is the 24%. Okay. So in this slide, I will explain the RNA viruses. What is RNA virus? So the RNA virus has the RNA instead of the DNA as their genomes. So what is a famous example of the RNA virus? The famous example is HIV, or the COVID-19 is also RNA virus. So RNA virus has the reverse transcriptase. So this is the reverse transcriptase. So we will see how reverse the transcriptase act. Okay, so after virus is attached to the host cell, and after that the viral core enter to the host cell. After that the viral DNA is also can enter the host cell. So after that the reverse transcriptase is bind to the RNA, and after that they made the complementary DNA. So it means that complementary DNA means the cDNA. After that the viral RNA is degraded. So also the reverse transcriptase can synthesize the second DNA strand. So they will make the DNA based on the RNA. Initially, the HIV the genome was the RNA, but they have the reverse transcriptase, so they synthesize the cDNA, and after that they will make the complement. They will make the double-stranded DNAs like this. So after that, this viral DNA can enter the nucleus and can be integrated into the host chromosome. So after activation, this viral DNA is transcribed into the viral RNA. After that, they can be translated. After that, they make their own protein, After that, finally, the assembled virus is birthed from the cell membranes like this. So this is the RNA virus. The very famous example was HIV or COVID-19. So HIV is RNA virus, and also HIV is also evolved from simian immunodeficiency virus, which call it as a SIV virus, found in the chimpanzee. So, usually the HIV virus, which is found in the human, is evolved from the SIV chimpanzee. So after that, this can become the HIV group 1 and group N and group O and group P. So usually the HIV attack the helper T cell. The green is this is electron microscope to show that T cell is green and the HIV is orange. So usually the HIV is attack the T cells. So in this slide, I will briefly explain the influenza. Influenza is also RNA virus, and also the rapid changes occur through genetic recombination. So main type is influenza A and B and C, but most cases influenza A. So influenza A is divided into the subtype based on the expression of hemagglutinin. Hemagglutinin, this is the purple is hemagglutinin, and the other yellow is neuroaminidase. This is Na, and this is Ha. So this table 9.5 show that strain of influenza virus A, so this is influenza A, responsible for major flu pandemics. In 1918, there was Spanish flu. The strain was H1N1. In the 2009, also there was a swine flu. It was the H1N1, but in 1968 there was a Hong Kong flu. At that time the subtype was H3N2. Okay, so in this slide I will show you the new strains of influenza virus. So this is the new strains of the influenza virus. This is the created by reassessment of the genetic materials from different strains. For example, the new H1N1 virus, swine flu virus, that was appeared in 2019, contained genetic materials from the avian virus, and also they also have the swine virus, and they also have the genetic materials from human virus. So it made the new strains. So in chapter 9 we learned about bacterial and viral genetic system. First, we learned about bacteria and virus have an important role in human society and the world ecosystem. Now, second, we learned about some genetic analysis of bacteria, and also we learned about some gene exchange through conjugation and transformation and transduction. And also we used the virus. We learned about virus. Viruses are simple replicating systems amenable to genetic analysis. So today we will finish the class.